Hello students, in today's session we are going to discuss about how the gene expression is regulated in your chapter Molecular Basis of Inheritance. Through these days we were discussing about the central dogma, starting with the DNA replication, then goes with transcription, then comes the translation. We have ended with translation. And by the end of the translation, we have manufactured proteins. So from all these processes, we have come to know one very significant point is that this is all very complex processes, multi-step plus complex processes. But when the processes are very complex, we need a perfect planning. We need a perfect and the systematic regulation. So here also DNA needs to be regulated. Now we will go for how this blueprint of our life, the major genetic material DNA, gets its own regulation. It is a self-sufficient system. There is nothing from outside to control DNA. DNA is the sole controlling part. So there are some special mechanism or a self-regulatory mechanism by which the entire process of DNA's functional role is getting regulated. That is what we are going to learn through today's session. When this regulation, when we study, we will go for two organisms like prokaryotic as well as eukaryotic. So in the case of prokaryotic organisms, the regulation will be taking place at the time of transcription. In prokaryotes, the regulation will be during transcription because they do not have any special segregation of cytoplasm, nucleoplasm, etc. So, because of this reason, they have only one site of regulation, that is initiation of transcription. But when we come to the eukaryotic system, eukaryotic cells, this gene regulation takes place in 4 levels. There are 4 distinct phases or levels at which the DNA regulation takes place. The first level is at the time of the formation of primary transcript of messenger RNA, that is taking place in the transcription. So here the primary transcript, that means hnRNA, we have already studied. hnRNA formation is the time at which the regulation will begin. That is nothing but transcription. The second event of regulation is that when this messenger RNA leaves the nucleoplasm and reaches to the cytoplasm, that time also regulation can happen. And one more thing is that at the time of eukaryotic transcription, they require some sort of processing. So the second is at the time of processing. If you remember, processing means splicing, capping, tailing, all these are at the time of processing. So processing is the second event during which the eukaryotic gene regulation takes place. And the third already we have seen, when this messenger RNA, after forming all these processing, it leaves the nucleoplasm and comes to cytoplasm for translation. And the fourth event is the normal translation. So we can see that there are 4 levels at which eukaryotic gene regulation takes place. Number 1, at the time of primary transcript of messenger RNA formation or hnRNA formation takes place during transcription. Second, processing or splicing. Then comes leaving the messenger RNA from the nucleoplasm to cytoplasm, and the last one is at the time of translation. Now, when scientists started studying about this gene regulation, there were 2 scientists worked for this very purpose. They are Francis Jacob and Jacques Monod. They were in 1961 doing their experiment in Pasteur Institute in Paris. Their experimental material was the E. coli, the prokaryotic E. coli bacterium. And even in 1965 they have got Nobel Prize also. So they have cultured E. coli in a petri dish. So when these prokaryotic E. coli were cultured, they want to know that at what stage this gene present inside the E. coli is getting regulated. They had given the substrate or food materials to this E. coli culture. The material was lactose. So you might have seen that these bacteria like the sugar as the food or the medium. So their favorite food is glucose, but if glucose is not available, they will go for a system of this controlled system for the digestion. Now Jacob and Monod gave lactose a disaccharide as the food. So this disaccharide they want to digest. We are all knowing that digestion need enzymes. Enzymes are nothing but protein. So they have to synthesize the protein. Like the protein synthesis, normal protein synthesis, they are preparing proteins in the form of enzyme. So this lactose is now digested into the galactose, their monomeric unit, monosaccharide, galactose and glucose. So for the purpose of the conversion of lactose into galactose and glucose, there comes the gene regulation, fully planned gene regulation. We are knowing that when we go for any kind of planned process, complexity will land up in perfect planning. So planned processes, you require more and more error-free steps or procedure. So let us see how come this gene is regulating the process of the breakdown of lactose. Whenever the word regulation comes, you will see that regulation involves some systematic steps. Like, for example, fan. Fan has got a regulator. Other than the switch, they have regulator. With the regulator, we can switch off. When it turns to 0, you are turning the knob to 0, you are switching off in a special way. When you increase the speed, you can just turn the knob, and finally if you want the maximum speed, you can regulate to fifth position or like that. So that means there is a system to switch on, switch off, or regulating the speed. Here let us see how this kind of regulator takes place. For that, these scientists have proposed a concept. The name of the concept is Operon concept. By the way, what is this term Operon stands for? Operon we use for a self-regulatory segment of DNA. Look at this segment, a piece of DNA from starting from here till this end. That is a very defined and a self-regulatory segment. Now the thing is that what is the meaning of self-regulatory? They will regulate themselves. They are automatic in such a way that they can switch on, they can switch off, they can proceed the processes. Everything will be under the control of this particular segment. So that specific segment, which is self-regulatory part of the DNA, is what we call operon. So operon, when they had analyzed, they could see that there are so many cistrons, so many segments. Segment number 1 P, segment number 2 I, all are represented by alphabets. Segment number 3, again P. Segment number 4, the O, right? Then so many segments like Z, Y, A. So all these segments will work in such a systematic and coordinated way. As a result, the whole protein synthesis will be perfectly taking place in a regulated way. Now let us see what are these alphabet. In your textbook also, page number 117, this picture is given. You can see in this picture two diagrams are given at the upper part and at the lower part. These two diagrams we have to learn because the two diagrams are showing two distinct phases of this gene regulation. Now let us see what is actually these alphabet stands for. P stands for promoter gene. We have seen promoter genes are the sites of RNA polymerase attachment. RNA polymerase is very essential here because RNA polymerase can only polymerize or synthesize messenger RNA for transcription. This messenger RNA will only go for the translation. So, RNA polymerase will fix at P site. Then you can see at the left side one alphabet I. I stands for inhibitor gene. So you can see there is a special process taking place. This part is very important for your content. So there is an inhibitor gene. This is also called regulator gene. But I stands for inhibitor. In your textbook it is written regulator gene. Both are correct. So this I stands for regulator gene as well as inhibitor gene. Now what kind of inhibition or regulation are they doing? This particular gene can produce a small piece of messenger RNA by transcription. This messenger RNA will be translated into a protein, and this protein is the real inhibitor protein for our gene regulation process. This inhibitor protein can also technically called repressor protein. Both the words stands for same meaning, repression, inhibition. Both will prevent something. Let us see what is that something prevented by this repressor protein. So once again, I stands for inhibitor gene or regulator gene, and this inhibitor gene will transcribed into a messenger RNA. This messenger RNA will be translated into a protein. We call it as inhibitor protein or repressor protein. In some reference book it is also written that accessory protein, which will play a vital role. Now we can see O, operator gene. This is the gene which can control the complete further process. What is this further process? Transcription and the formation of messenger RNA, translation, etc. Just like operation for any kind of industry, there is a site where the real operation takes place. The real production, manufacturing takes place, which will have some machineries and all. There the technique is really done for the final output. Like that, here is the operator gene. And then comes three structural genes, SG, structural genes. LAC-Z, LAC stands for lactose. LAC-Z, which can transcribe into messenger RNA and finally make a protein in the form of the enzyme beta-galactosidase. Then comes LAC-Y, which will transcribe and translate into another enzyme permease, and a third structural gene, you call it as LAC-A, which will transcribe into a third protein, you call it as trans-acetylase. By the way, what are the functions of these enzymes? Beta-galactosidase help in the conversion of lactose into glucose and galactose. The real breakdown, right? The way amylase work in our body, protease work in our body, lipase work in our body. For this E. coli, the beta-galactosidase will be the real breaking enzyme. digesting enzyme. It can digest the lactose into the galactose and the glucose. Once galactose and glucose is forming, easily they can get the energy. And what is permease? As the name implies, as permitting. What is permitting? Permission of lactose to enter inside the cytoplasm of the bacteria. Bacteria has got a very tough cell wall. So cell wall will prevent the entry of lactose. Until and unless this lactose is getting inside, they can't go for digestion. So permease enzyme will only allow the entry of the lactose into the cytoplasm. So permease function, beta galactosidase function is all over. Now we come to transacetylase. So far transacetylase function is not known. So I have explained each and every segment of this operon, segment of this particular gene, or what we call DNA segment. Now let us see how the real action happens. There are two modes for this operon. One mode is what we call switch off mode. Switch off mode. Switch off mode means no action. No any kind of protein production takes place. Everything will be in a off mode. Let us see how this can perform an off mode. That means without any kind of protein production. When this operon will be getting an environment without any kind of food. If food is present, then only we require to prepare the enzyme. Otherwise, no need. Otherwise, why we waste our energy for the synthesis? So, when you don't give them a pinch of lactose in their culture medium, this switch off mode will be attained by the operon. How it will be switch off? You can see this repressor formation takes place. That is point number 1. The inhibitory protein or regulatory gene prepare messenger RNA. Messenger RNA will produce repressor protein. This repressor protein is acting like an inhibitor. What are they going to inhibit? They will go directly to the operator gene. Look at the operator gene, and they will immediately block the operator gene. When the operator gene is blocked, naturally what will happen? All the operation will be stopped. Then how it will be stopped? Our RNA polymerase cannot proceed towards the structural gene. Because RNA polymerase reaching to structural gene is very essential for the polymerization of messenger RNA. When this proceeding is not happening due to this blockage of the repressor protein acting like an inhibitor at the operator gene site, no movement of this RNA polymerase. So no transcription, thereby no translation. Switch off mode, right? Now we have to make it into switch on mode. How can we make it into switch on mode? So we will go for how it can turn on. On position can be attained when you provide them a pinch of food, the lactose. When lactose you are giving, suppose this is the food lactose, right? One small granule of lactose, maybe 100 granules of lactose. Out of that, few granules will act for this, permease will be allowing these granules to get inside. So what will happen? This lactose will combine with this repressor. At this point of time, this lactose molecule will combine, will hold the repressor molecule, thereby this repressor cannot reach to operator site. That is something like you have got a troublesome person. You are 100% sure that this troublesome person will go and give some sort of unwanted messages so that the work will not be done. The work will be hampered. The same thing happens here. So what you can do, you stop that fellow on the way. The same principle is happening here also. The repressor protein will be caught hold on the way, so that this inhibitor will stop the movement of the repressor protein and blocking the work of this operator, and as a result what is happening, operator is now exposed, operator is now free. When operator is free, you can expect the RNA polymerase will freely proceed towards the structural gene. So structural gene will get the signal, structural gene will start producing this messenger RNA. When messenger RNA is transcribed, you can expect that translation can happen. When translation happens, the particular protein in the form of enzymes can be manufactured. Everything is perfect. That means it is in a switch on condition. So here who had given that switch on induction, or it is almost like an induced by one food particle. What is that molecule? Lactose. So lactose is here acting like an inducer. So this lactose will induce the complete operon to switch on, and everything will be perfectly done without any delay. So finally you have prepared 3 proteins in the form of 3 enzymes, and very easily the E. coli can synthesize the glucose as well as galactose from the lactose. This is the way the gene regulation takes place in the prokaryotic level. Now they have switched on. Now we have to again switch off. How it can be turned again to switch off? All the lactose is completely broken down. So when this lactose is broken down, you have got all the 100 molecules are finished. So no more lactose to stop over here. So when this lactose is completely used or completely digested, the repressor is now very strong. The repressor will immediately reach to operator site and block the operator. Again the complete operon system is switched off. So this is the way the operon is getting switched off and switched on. In your textbook, 2 diagrams which I have already told. The upper one is switch off mode and the lower one is switched off. I have made it in this single diagram, making it switch off as well as switch on. So children, this is the way the gene expression by a self-regulatory mechanism is working out in the genetic level. So from this you can understand that the complex systems, whether it is a replication, whether it is a transcription, whether it is all about the translation, everything is perfectly taking place in the system. I hope that all the points today which we have learned is clear for you, and we will be getting into next topic in our next class. Thank you.